Okay, so what is a test cross in genetics? So let's say that there is a person who has a dominant phenotype of brown eyes, but we don't know their genotype by just looking at them. Because if you have the dominant phenotype, that means you have at least one dominant allele for that phenotype, but you don't know if you're homozygous dominant or heterozygous. But if you see a blue-eyed person who has the recessive phenotype, then yeah, you would know that they are homozygous recessive because that's the only genotype that would code for that phenotype. But now let's pretend that this person was adopted and they don't know what their parents' phenotypes or genotypes, their grandparents, they have no idea their family history of eye color. So this person, he has the dominant phenotype of brown eyes. How would we find out his genotype? Well, nowadays we do have DNA sequencing, like you could do 23andMe and find out if he is homozygous dominant or heterozygous. But if that's not an option, we have what's called a test cross. So in a test cross, you actually cross, this is like the definition, you actually will cross a dominant unknown genotype. So you can tell they have the dominant trait by looking at their phenotype, but you don't know their genotype. You would cross them with a homozygous recessive. And so in a test cross, you cross an unknown dominant genotype with a homozygous recessive. So here, this capital B would represent the male here with brown eyes. We know he has at least one dominant allele, but we don't know his second allele. Whereas the blue-eyed phenotype, female, we know is homozygous recessive. So we could expect that some of their children will have brown eyes, but we don't know the second genotype option here. So if they mate and have a bunch of children and all of their children have brown eyes, then we can infer that this brown-eyed person, his genotype is homozygous dominant. If 100% of the F1 or 100% of the offspring have brown eyes, then, and if it's only like 2 kids, then maybe not a large enough sample size to confirm. So when you see test crosses, hopefully the amount is a lot to draw conclusions from. So here I said like 8 kids because that's a little bit more reliable, that the chances are that he is homozygous dominant. If every single one of them, they only ever make brown-eyed children. Now, let's pretend though that when they have offspring, some of them have brown eyes and some of them have blue eyes. Well, that tells us that the male with the unknown genotype is heterozygous. That would be the only way for this couple to have some children with brown eyes and some children with blue eyes. So we can confirm that his genotype is heterozygous. So I do want to point out, though, that just because you see here 2 out of 4 or 50% likelihood of brown eyes and a 50% likelihood of blue eyes doesn't mean if they have 8 kids, 4 have to have brown and 4 have to have blue. This is just every time they have an offspring, there's a 50% chance of brown eyes, a 50% chance of blue eyes. So if you're ever looking at data, it might turn out, let's say they have 8 kids, maybe 5 have brown eyes and 3 have blue eyes, or 6 have blue eyes and 2 have brown eyes, right? Like it doesn't have to be 50-50. All right. So again, to summarize, a test cross is used when you cross a dominant phenotype of unknown genotype with a homozygous recessive, and then you look at the offspring, and based on the phenotypes of the offspring, you can determine if that dominant parent was homozygous dominant or heterozygous, depending on what the offspring looked like. All right, good job.